So this is what happens in a non-myelinated fiber. So in a non-myelinated fiber, an action potential has to be generated at every single point along the axon in order to prevent those leakage channels from causing the voltage or the local current to decay. So we have to regenerate an action potential at every single point along the axon. So we can see that here. So in response to a signal, we're going to have that first region of the axon depolarizing. There's going to be this kind of wave of depolarization that's going to trigger the adjacent membrane to be depolarized, reaching threshold, initiating another action potential at this region. While this region is now depolarizing, that previous region is now repolarizing. So this is going to continue as this wave of depolarization spreads in one direction along that axon, regenerating that action potential at every single point. So in a non-myelinated fiber, an action potential in one segment causes the membrane in the next segment to come to threshold because of the influx of sodium ions. So an action potential is going to be propagated from one segment to the next, or at every single segment along that axon. Once again, that has to happen because otherwise that wave of depolarization would decay because of the leaky plasma membrane. Once again, an action potential is propagated in one direction, so it begins at the axon hillock and it goes all the way down to the axon terminal. Propagation of an action potential in a non-myelinated fiber is called continuous conduction. So continuous conduction refers to the propagation of an action potential in a non-myelinated fiber. In a myelinated fiber, we have that myelin sheath wrapping around the axon. That provides, or that the myelin sheath functions as an insulated covering. So we can see that here. We've got this myelin sheath wrapping around the axon. We've got these regions where we don't have the myelin sheath, known as the nodes of Ranvier, or our myelin sheath gaps. In a myelinated fiber, we actually only have to regenerate the action potentials at the nodes of Ranvier, or the gaps in the myelin sheath. So in a myelinated fiber, the action potential is generated at the axon hillock, and then it's going to jump from one node of Ranvier to the next. The reason for that is because the myelin sheath helps prevent the leakage of those sodium and potassium ions because it's an insulated covering. So in other words, it prevents that wave of depolarization from decaying with distance. So we're not losing a bunch of ions in these myelinated regions due to those leakage channels. So that means that wave of depolarization can very quickly spread from one node to the next. So action potentials only have to be regenerated at these nodes. So that causes the action potential to essentially jump from one node to the next. So at this node, we might see that action potential regenerated, which means you have voltage-gated sodium channels opening. So that causes a bunch of sodium to flood this region of the neuron. Those sodium ions are then going to be attracted to the adjacent membrane that is more negatively charged. So that creates this wave of depolarization. That wave of depolarization can shoot right across this myelinated region to the next node. Then that depolarization can then trigger an action potential at this node. So we're then going to trigger the opening of voltage-gated sodium channels at this node, causing depolarization and another action potential. So because of the myelin sheaths, we actually only have voltage-gated sodium channels located at those nodes of Ranvier. So because the wave of depolarization is capable of spreading all across the myelinated regions, we only have to regenerate action potentials at the nodes. So therefore, we only need those voltage-gated sodium channels at the nodes of Ranvier. So in a myelinated fiber, action potentials are spread along the axon in what's called saltatory conduction. So in saltatory conduction, that action potential is jumping from or leaping from one myelin sheath gap or one node of Ranvier to the next. Once again, action potentials do not need to be regenerated in that myelin sheath region because the myelin sheath acts as an insulated covering preventing the leakage of ions. So therefore, that wave of depolarization does not necessarily decay in that region. So we only are going to need those voltage-gated channels at those nodes of Ranvier. So the sodium that enters at one node is then going to be carried essentially to the next node in order to trigger depolarization of that next node, bringing it to threshold, triggering another action potential. So in non-myelinated fibers, conduction is going to be really slow. So continuous conduction is extremely slow in non-myelinated fibers because that action potential has to be generated at every single segment along that axon. In a myelinated fiber, or during saltatory conduction, the action potentials only have to be regenerated at the myelin sheath gaps or the nodes of Ranvier. That means that action potentials can readily jump from gap to gap, meaning that conduction is going to be extremely fast. So in non-myelinated axons, conduction is going to be very slow. This is known as continuous conduction. Again, that's because that action potential has to be regenerated at every single point. In myelinated axons, conduction is extremely fast. It's also known as saltatory conduction, where that action potential is jumping from one myelin sheath gap to the next. Propagation speed is not only impacted by a myelin sheath, but it's also impacted by the diameter. So the presence of a myelin sheath is ultimately going to increase the speed of conduction, whereas the size of the diameter, so the larger the diameter, that's also going to increase the speed of conduction. So the larger the diameter of that axon, the less resistance to flow, or the less resistance to that current. The reason for that is, if you think about it, the larger the axon, there's just more space for all of the cellular components. So that means there's going to be less obstructing the flow of those ions from one region of the plasma membrane to the next. So there's going to be less resistance to that wave of depolarization in larger axons. So if you've got a myelin sheath, that's going to increase the speed of conduction. The larger the diameter of the axon, that's going to increase the speed of conduction. So motor neurons are an example of a neuron that has an extremely fast propagation speed. So our motor neurons have a very large diameter, and they're typically myelinated, which means they have a propagation speed of anywhere between 15 to 150 meters per second. So that's extremely fast. Some of our pain afferents, on the other hand, so our neurons that sense pain, actually have a somewhat slow propagation speed. So they have a slow propagation speed because they have a smaller diameter, and they tend to be non-myelinated. So our pain afferents have a propagation speed of about 1 meter per second. So now we're going to watch one of our A&P Flix videos on the propagation of an action potential.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.